Okay, so in this lecture, we're going to look at what I've called wiring and plumbing. So essentially, it's how tubes are made in the body. And this is a process called branching morphogenesis, and how the nervous system is allowed to develop through the body, and that's called axon guidance. So we're going to look at branching morphogenesis, axon guidance, and vasculogenesis, the formation of new blood vessels. So the lecture plan is first of all to understand why there needs to be a process of branching morphogenesis. Then we're going to look at basic concepts in branching morphogenesis. We'll look at the mechanisms of morphogenesis, the signaling pathways that allow branching morphogenesis to occur. Then for our wiring, our nervous system part of the lecture, we're going to look at repulsive and attractive cues, how axons find their way around the body using something called the growth cone. And then we're going to look at a process of lateral inhibition, which is required for both branching morphogenesis and axon guidance. And this involves our old friends Notch and Delta. So why do we need branching morphogenesis? And a way to think about this is that all organisms are three-dimensional objects, but all exchange of gases and materials occurs across a two-dimensional surface. And that's okay when the organism is small, like a single-celled organism. Then it can just take everything it needs through its main membrane. However, after a certain size, and actually this isn't too many cells, this doesn't work because you can't get the materials, the food, the oxygen into the cells you need to. So in a higher animal, exchange surfaces have to be folded within the organism to vastly increase the surface area for exchange. So here's our hypothetical single-celled organism, and then here is the branching morphogenesis of the alveoli of the lung. And you can see here that this branching and multiple tubes makes a much more complex structure which has a vastly increased surface area for gaseous exchange. So lungs are a product of this branching morphogenesis. So for example, gaseous exchange in the human lung, in our lungs, occurs across a surface of around 300 million alveoli with a total surface area of about 100 meters squared, which is approximately the size of a tennis court. So all of that is folded up inside your lung. And this is all produced by this process of branching morphogenesis. So branching morphogenesis can be thought of as a kind of program by which there are multiple stages. There's the initiation of a bud, and then that bud can be extended, and then possibly at the end of that bud we can have a split. And so of course this can be an iterative process, by which I mean we can have initiation of a bud, extension of a bud, splitting of a bud, but then we get extension again of a bud and splitting of a bud. And this increases the surface area, increases the complexity of the structure. And the way this is iterative, we can get highly detailed, highly complex branch structures within a certain space. And one of the classical models to look at this process of branching morphogenesis is the Drosophila tracheal system. So Drosophila don't have lungs. They have a series of spiracles, which are essentially holes in their outer body to take in oxygen. And it's transported then, the oxygen, through these spiracles by the tracheal system. And here in figure 1, we can see a stain for the whole of the tracheal system. And you can see here we have a major tube, and then we have all these branch tubes off of it. So these Drosophila larvae show all the programs we were discussing previously. We have instances of initiation, here extension, branching, and then increased branching from those extensions. So we're showing not only those 3 programs, but we're also showing the iterative nature of the development of the Drosophila tracheal system. So, as you might expect, it being Drosophila, there are many Drosophila mutants that impair the formation of this tracheal system. And one of these is called trachealess, i.e. no trachea in the mutant, and this is the master regulator of the trachea and is expressed prior to trachea formation. So we can see here in our wild type, we can see that trachealess mRNA is expressed in our segments prior to the formation of the trachea. And then we can see here that the RNA, so this is an in situ hybridization, we can see the RNA for trachealess is being expressed in this region here, and you can see the tubes being produced in a later stage of Drosophila development. So these trachealess mutants have no trachea structures. So all of these genes are markers of specific parts of the trachea structure, and we can see here these are our trachealess mutants. Here's the wild type. On this side we have a very good example here. This OSI15 gene marks the whole of the tracheal system, and in our trachealess mutants we don't see any trachea structures. And this holds true for any of these genes which are required to form parts of the trachea structure. So all of these genes are downstream of trachealess, and when we have a mutant trachealess, they no longer express these genes. So to recap, the trachealess mutants have no trachea structures. So what is trachealess? As you might expect from this extreme effect of trachealess, and it being expressed actually quite early on during development, it is in fact a transcription factor. It's a basic helix loop helix Paz domain protein. So the important question to ask is, what are the main targets of this transcription factor? And of course, the previous experiment alluded that possibly all of these genes are downstream of trachealess and therefore may be targets of trachealess binding to DNA. So 2 of the key downstream targets of trachealess are 2 genes called breathless and branchless. And the mutants of these genes have identical phenotypes. Both of these genes are downstream of trachealess. So here we have our wild type, the A and C panels here, and on the right hand side we have a branchless mutant, and we can see here we no longer get that elimination of the trachea structures in their totality. Instead we get a vastly curtailed tracheal network. So the difference in our more developed trachea network is quite clear. We see no more of this lovely branching from a central trachea pipe, if you like. So the branchless mutant has an impaired trachea formation. And we also see this in the breathless mutant. It's more or less identical. And again, this is a dye that marks where the trachea is within the larva. And you can see in a very similar fashion to how the branchless tracheas looked. The breathless trachea looks very similar. We don't see any extension of these tracheal buds. So what are breathless and branchless? They must be highly related to each other, at least functionally, because their phenotypes are virtually identical. And so it turns out, branchless is a ligand, it's a secreted protein, and breathless is the receptor for branchless. And what we can see here is that wherever there is a branching process occurring, we see this functional interaction between cells that express branchless and cells that express breathless, the receptor. So we have this reciprocal interaction. Branchless binds, breathless is bound by branchless, and we can see this at any stage where we're requiring this extension and branching to occur. So we know that branchless is a ligand, and we know that breathless is a receptor. But what is the molecular mechanism of these? What are these proteins in actuality? So we've touched on the fibroblast growth factor. We looked at that as part of the clock and wavefront model in somatogenesis, and we touched on it briefly in our signaling toolbox lectures. But essentially, fibroblast growth factor binds the fibroblast growth factor receptor, which is a typical growth factor receptor. And as we know, growth factor receptors control cell proliferation, cell survival, and cell motility. They control cell proliferation through well-known, and we've discussed this before, the RAS pathway, and through mitogen-activated kinases, we can induce cell proliferation. Cell survival is induced through a different kinase pathway. This is called so-called AKT pathway, and cell motility is typically controlled through a secondary messenger system, through calcium and the roles of calcium in the body. So calcium signaling can be one of the outcomes of fibroblast growth factor signaling. So you can make cells move in space, they can make cells survive, and more importantly, they can make cells grow. And all three of these functions are required for the branching and elongation processes that are occurring during trachea formation. So the next step of our trachea formation is sprouting. And unsurprisingly, there's a mutant that has defective sprouting called Sprouty. And here we're again looking at the trachea network. In our wild type, we see this very nice arranged pattern of branches, whereas in our Sprouty mutant, we see a much more disorganized and overproduced tracheal network, many more branches. This suggested to the people working on this mutant that Sprouty must be an inhibitor of branching. And so it turns out, Sprouty is a secreted inhibitor of fibroblast growth factors. So remember, the fibroblast growth factor, branchless, is secreted. This gene, Sprouty, can inhibit its function by preventing FGF binding the FGF receptor. This is the first cysteine-rich protein ever discovered, which is a large family of soluble inhibitors of fibroblast growth factor family proteins. And actually these cysteine-rich proteins and proteins that contain things called cysteine knots, genes such as Cerberus, are also a member of this family. And this gene, Sprouty, is expressed at the tips of the trachea branches. And we can see this at the different stages of formation. So here is our growing tip. We can see expression of Sprouty by in situ hybridization here. And then we get these regions of Sprouty that are expressed where where trachea branching is required to be inhibited. So Sprouty inhibits the signaling caused by branchless and breathless. So we have this concept of activating the pathway and also the mechanism to dampen it down to be able to finally control the amount of branching in the Drosophila trachea. And then the end stage is a mutant called prune. And in case you didn't know, to prune something is to remove unwanted growth. So you typically prune a plant to get rid of growth and allow it to grow again the next season. And the end stage in branching morphogenesis in the trachea is what's called terminal branching, because we don't want that iterative process of elongation and branching to happen continually. We need it to stop. So how does trachea formation stop in Drosophila? Using, and we can find this out with this mutant pruned. So what we require is no more growth of cells, and that cell to then terminally differentiate by becoming a branch cell. So let's see how this works in practice. We have our primary branching, and these branches are caused by elongation and increased cell division. So we get those cell movements that we've talked about before, the intercalation and convergent extension and epiboly, and together these processes elongate and cause branching of the main trachea. But finally, and we can see in this figure here, we get a final cell that terminally differentiates into what's called a terminal cell, which itself is highly branched but cannot divide and cannot undergo the rest of this program. So with these cells, growth is stopped and we get terminal cell branching. And we can see the effect of pruned in our pruned mutant. So when we have normal pruned expression, we get a final cell that has terminally differentiated and forms all these fine processes. In contrast with our pruned mutant, rather than getting a final terminal cell with all these fine processes, we in fact continue on this process of branching by cell division and iteration of formation of branches. So this pruned mutant continues to go through that program, but our wild type with the active pruned gene allows it to stop and terminally differentiate. And you can see that here in this circle I said before, but that circle shows one of these terminally differentiated cells with all of those final processes. And this cell can no longer divide. So what is pruned? Pruned is the Drosophila homologue of a protein called the serum response factor, otherwise known as SRF. And this is one of these transcription factors that's downstream of growth factor receptor signaling. So we have our growth factors, they bind our receptor tyrosine kinase, typically through RAS signaling. We get activation of ERK, which is one of these tyrosine kinases, and then this protein kinase enters the nucleus and activates SRF. And serum response factor then activates programs of motility and terminal gene expression in cells where it's activated. So what does serum response factor do? Serum response factor can change epithelial cell shape, and typically serum response factor allows the formation of processes called filopodia. So in a cell, filopodia are kind of spiky outgrowths of the cytoplasm. And in a knockdown of this serum response factor, these filopodia are no longer made. What does serum response factor do? It can control the structure of the actin cytoskeleton in the cell. So in our wild type cells that are growing and moving, we can see these very well arranged filaments of actin, and filamentous actin or F-actin can be detected using the toxin phalloidin. So using phalloidin and using microscopy, we can see the actin cytoskeleton. When we knock down the serum response factor, we see much less organization of these fibers that are called stress fibers. And one of the reasons they're called stress fibers is that when cells are starved of serum response factor then gets activated, and one of the first effects you see in stress is the formation of these stress fibers. So that's how serum response factor was discovered, and true to its discovery, serum response factor can control the expression and localization of actin within a cell. So therefore, without serum response factor, we can't produce those cells that have terminal processes in them, and therefore the cells can continue to proliferate and grow. So we have our overall pathway of tracheal morphogenesis, where the first thing is specification of cells that are going to become the trachea through that basic helix loop helix transcription factor, tracheolis. So we get the initiation of the first tracheal sac through the expression of tracheolis. Then we have that interaction between the FGF receptor or breathless in the tracheal epithelium and branchless, which is secreted by nearby cells, which is our fibroblast growth factor secreted factor. And this reciprocal interaction allows cells to divide and extend. Then remember we have that gene sprouty or pointed, and sprouty allows for the splitting of the branches by cell division. And then finally we have pruned, which is able to, in response to branchless signals, so in response to growth factor signals, pruned serum response factor is able to change the shape of that final terminal cell to allow those branches to form. So you can see here that we use that same pathway, the FGF receptor and FGF signaling molecule, but being modulated by that downstream serum response factor pruned. We can either control growth and splitting of the tracheal tubes or their final terminal differentiation. And this is a highly conserved pathway. So human lung morphogenesis does exactly the same thing. It requires FGF10 and FGF2b, as well as Sprouty2, which is the mammalian homologue of Sprouty, and together these work to complete lung morphogenesis. So we have our bud induction, where we have the FGF from the distal mesenchyme activating the primordial bud in the same way that we get the initial sprouting from this reciprocal interaction in Drosophila. We get elongation, which requires this continued interaction of FGF and its receptor, as well as the expression of sprouty, which allows that splitting to occur. And then we get that final differentiation, again using FGF signaling, but again through transcription factors that will, such as serum response factor, that allow for that final cell fate, that terminally differentiated fate of the lung alveolar. So although there's more complexity in the human lung, the central signaling between FGF and the FGF receptor is highly conserved, as is the use of sprouty and serum response factor to form branches and terminally differentiated alveoli. So let's move on to this concept of attractive and repulsive signaling. So for branching morphogenesis to be effective, there needs to be a way to prevent branches from collapsing into each other. There's nothing to stop them from doing that. So there needs to be a signal that tells the branching developing trachea or lung not to go into a certain area of the developing embryo. And this process is called repulsive signaling. Conversely, to make a complete vasculature or other tubular network, sometimes the growing regions must be attracted to each other so that we can make sure that there's enough of a vasculature, enough of a tracheal system in that area of the developing embryo. And this process is known as attractive signaling. And both of these occur during a process called axon guidance. During central and peripheral nervous system development, both attractive and repulsive signaling is required. A neural crest and neural tube neurons can migrate their processes, so making the peripheral nervous system, which is one of the major functions of the neural crest and neural tube neurons, they have to move very long distances to be able to perform their function. So let's revise the structure of a neuron. So we have the dendrite containing the cell body. Then we have an axon, which ends in an axon terminal, and both these are required for innervation, one in the central nervous system and the other one at the peripheral tissue. But it's the axon itself that migrates to the region that requires innervation. And just to be aware, the axon has to migrate a very long distance. Typically, it has to migrate from the central nervous system to the furthest extremities of the body. So in some cases, that requires the axon to migrate in excess of 1 meter. And do be aware that that's a single cell that's doing that. So how can a single cell be able to migrate that distance without bad things happening to it? How does the migrating axon know where to go during development? So here is the business end, if you like, of the axon. And this is called the axon growth cone. And if we have a look at key parts of its structure, the microtubules and actin, you can see that there's these actin spikes all over the place at the top of this axon growth cone. And you can see that the microtubules that are quite organized in the axon become more disorganized in the growth cone. And the implication here is that that growth cone is very motile and can throw out actin processes and can change its shape quite quickly due to the changing cytoskeletal elements within it. And as we know, and I would have asked you this question, but we can't do that using this method, but we all know that those receptors that allow cells to bind other cells expressing the same receptor are called integrins. Oh, sorry, cadherins. My apology. Cadherins. They're called cadherins. And how do these cadherins play a role in this axon guidance process? Well, there's a process called fasciculation. And fasciculation is the key attractive cue that makes numbers of axons become nerves. So in this process of fasciculation, axons are attracted to each other, and they do this through the homotypic receptor N-cadherin. So here we have this process of fasciculation where, due to the expression of N-cadherin, these axons are attracted and bind to each other, and their growth cones migrate together. And then once they get to the tissue that they need to innervate through a process called defasciculation, the removal of the expression of N-cadherin, then these growth cones and the axons behind them then separate from each other and are able to migrate through that terminal tissue destination individually. And as I said, this requires the control of the expression of this protein N-cadherin. So growth cones themselves can be attracted towards or repelled from specific areas of the body. And how does it do this? There are specific attractive cues and specific repulsive cues that can activate or inhibit these RAC and Rho kinases. And these Rho kinases, for instance, make Filipodia by remodeling actin. And we can see this here in this schematic where there is active Rho kinase function. We see the expression of these lamellipodia and Filipodia, and this allows that growth cone to migrate through the tissue. So what are the attractive and repulsive cues that are required for this axon guidance process to occur? And they come in two families. We have the ephrins, semaphorins and slits, which are repulsive cues, and we have netrins, which are attractive cues. And what are the effects of these on the growth cone? So here in this figure, we see the expression of actin in the actively growing growth cone. And if you express semaphorins and ephrins in the vicinity of these growth cones, you can see a very clear response. These Filipodia and lamellipodia are collapsed. The growth cone becomes much smaller and less motile. So both the semaphorins and the ephrins can collapse this axon growth cone. In contrast, if you express netrin, the effect on that growth cone is to increase that actin polymerization, that Rho-mediated actin polymerization. And we can see this in our control with a normal sized growth cone. When we treat with this protein netrin, we get increased size of the major part of the growth cone, so increased lamellipodia formation, and we also get increased numbers and size of these filopodia. So Nectarin-1 induces growth in that growth cone, and it also increases the polymerization of actin. So let's go back to some of the complications that there are in branching morphogenesis if you think about how the process happens. So we have our sac, we have primary branching, and then we have elongation and then branching at the end of these stalks. Now, why is it that only the cells at the end of the stalks can form branches? Why is it they form branches, and why isn't it that the red or green or yellow cells behind it can form branches? We haven't yet discussed the molecular mechanism by which that control occurs. How one cell can have one fate and the cell adjacent to it can have a different fate. And this is a process called lateral inhibition. And lateral inhibition is simply a process by which cells that can inhibit nearby cells from assuming the same fate. And this happens during nerve cell development in Drosophila. During early nerve cell development, we have unspecified epithelial cells. Then stochastically, one of these cells adopts a nerve cell fate. And then as soon as that cell does, the cells around it are inhibited from a nerve cell fate and become epithelial cells. And this is caused by the Notch delta signaling pathway. Remember, it's a juxtacrine signaling pathway, and it prevents two adjacent cells from having the same fate. And this is what's happening here. We have one cell in which delta signaling is active, and then in all the other cells, Notch signaling is inactive. So this is juxtacrine signaling, where there's competition between the cells and only one cell can win. So that all these cells have the capability of signaling Notch delta, but as soon as one of them actively signals, this cell becomes specialized and then inactivates its Notch signal so that other cells cannot adopt the same fate. And this is the key process of juxtacrine signaling by Notch delta. And we've seen this also in somatogenesis, where Notch delta signaling defines the limits of the developing somite. So again, we're seeing an example of how lateral inhibition by Notch delta is specifying a specialized cell fate at one cell at the expense of other cells. So delta inhibits the preceding cell, but the preceding cell can still grow and provide motive force. So we have the cells before the stalk growing. This cell is inhibited from, this cell inhibits the cells below it from adopting a branching fate, whereas this cell can adopt a branching fate. And that delta Notch signaling in the presence of branchless and breathless and downstream factors is what controls which specific cells are able to be those terminally branching cells. And if we inhibit this Notch delta pathway, this can cause oversprouting. So we can inhibit the Notch delta signaling pathway either by treating with a drug called DAPT or by making a genetically defective delta And here we're looking at the ordered growth of retinal blood vessels, which has to be quite well controlled so that there's a good blood supply across the whole retina. In our controls, we can see a nicely organized branching of the retinal blood vessels. However, when we treat with DAPT, or we look at a mutant with genetically defective delta, we can see that there's oversprouting occurring, and the clearly defined, nicely arranged system of blood vessels is lost because every cell is able to sprout and terminally differentiate. So we get a sort of shrunken system where every cell tries to adopt that final fate, and this causes disordered growth of the retinal blood vessels. So that's the end of this lecture. What did we cover? We covered the concept of branching morphogenesis and why we need to have branching morphogenesis to increase the surface area of a tissue for gaseous exchange or the exchange of nutrients. We looked at basic concepts in branching morphogenesis, this iterative program of growth, extension and branching. Then, using the Drosophila trachea as a model, we looked at genes involved in the mechanisms of morphogenesis, the fibroblast growth factor and the fibroblast growth factor receptor, as well as the specification by Trachealis and the serum response factor required to make the branching in the final terminal cells. We looked at repulsive and attractive cues. We looked at semaphorins, ephrins and netrins, and how the presence of these proteins through activation or inactivation of Rho and RAC kinases prevents or allows polymerization of actin and therefore movement of things like the growth cone that we see in axon guidance. We also looked at fasciculation, which allows axons to move together to a tissue site. And then finally, we looked at lateral inhibition and the role of Notch delta to specify the fate of one cell and preventing the cells adjacent to that cell from adopting the same fate. So that's it for this talk. We shall... Oh, there we go. Stop share. We'll come back at the hour. So just after 15 minutes, we'll have the next lecture. All right, I'll see you then.